Kendra has fallen ill with some kind of bacterial infection, and it's up to us to figure out what she's been infected with so that she can be treated properly. One way that we can identify the bacteria that Kendra is infected with is by determining whether it's aerobic or anaerobic. An organism's need and affinity for molecular oxygen can be assessed by growing the bacterium in thioglycolate tube cultures. When we used this method to grow our isolated bacteria, most of the bacteria was concentrated at the top of the tube, but there's some growth further down as well. Using this observation, we now know that the bacteria that's causing Kendra's illness is a facultative anaerobe. This means that the bacteria can grow with or without oxygen, but it grows best in the presence of oxygen. Another method that we can use to identify the bacteria is by growing it on MacConkey Agar. MacConkey Agar is a selective and differential medium that only grows gram-negative bacteria and differentiates between gram-negative bacteria that can metabolize lactose and those that cannot. When we did this, there was no growth on the MacConkey Agar plate. This means that the bacteria that is infecting Kendra must be gram-positive. Another test that we did to determine what bacteria is causing Kendra's illness is an acid-fast stain. Acid-fast stains are able to differentiate between two different gram-positive cells, those with and without mycolic acid in their cell walls. This is done by using carbylfuran as a primary stain, which is retained by acid-fast cells after the decolorizing agent is applied, and methylene blue as a counter stain, which turns non-acid-fast cells blue. In an acid-fast stain, acid-fast cells are pink. In this image, you can see that the bacteria that's affecting Kendra stained blue, which means that it is not acid-fast. The last test that we did to determine what bacteria has infected Kendra is a blood agar plate. Blood agar is a differential medium that tests bacteria's ability to produce hemolysins, which are enzymes that lyse red blood cells. There are 3 different degrees of hemolysis: beta, alpha, and gamma. From our blood agar plate, we can determine that our bacteria is beta-hemolytic because the area surrounding the colonies was white. To summarize our findings, the bacteria that's making Kendra sick is a facultative anaerobe, gram-positive, non-acid-fast, and beta-hemolytic. After a little research, we should be able to identify the bacteria. Based on our findings, we determined that the bacteria that's infecting Kendra is Staphylococcus aureus. Now Kendra can be properly treated, and those that she's come into contact with can seek medical attention as well.